Welcome back, friends. In this video tutorial we'll be talking about DNA methylation and the effect of DNA methylation in the gene transcription and obviously the expression of genes. Now what methylation can do is that they can silence gene by modifying the DNA and also by modifying histones. Now there are several type of methylation, there are several type of modification systems and processes available for gene expression. Methylation is one of them, and so as we are having acetylation, we are having phosphorylation, ubiquitination, and methylation is one of the very important one because it regulates the expression of gene, right? Now this DNA methylation, it's not right to exact right this DNA methylation, because methylation can be of two different type. One is a DNA methylation, another one is of protein methylation or histone methylation, right? So it can methylate DNA and histone both. And we are also going to see that DNA methylation or histone methylation, whatever type of methylation we're talking about, they are very specific about the methylation process. So they are having some specific sites present in DNA and also present in histone or protein sequences onto which they methylate, onto which the methylation usually occur. Okay, so let's begin. So first of all, it's about the methylation introductory part. This DNA structure can be modified by covalent attachment of methyl groups. Now I remind you, this methylation or addition of this CH3 group to either DNA or protein leads to covalent modification or covalent change. That means this change is irreversible because it's a very strong bond of covalent interaction. And this kind of methylation is very much abundant in eukaryotic systems, more importantly in plants and vertebrates. So you can see in higher eukaryotes we are going to find this kind of methylation in their DNA as well as in their histone. Okay, now this particular phenomena of methyl transfer is carried out by the enzyme methyl transferase. Now if you're talking about methylation to the DNA, we'll be talking at DNA methyl transferase, or if you're talking about histones, then it will be called histone methyl transferase. DNA methyl transferase is written as DNMT, DNA methyl transferase is written as DNMT, and histone methyl transferase is designated as HMT. So these are the two abbreviations of two different type of methylase. Now DNMT methylates DNA and HMT methylates uh, histone proteins. Okay, now here is the example of methylation. Now if we're talking about this is a DNA as a base, it is a cytosine. Now in this case we are having a methyl group. Now this methyl group will be donated to a particular position. Now I've told you that there is specific site for the methylation, and in this case suppose this 5 carbon is a perfect size for the methylation, so this methyl group will be added to this 5 carbon position onto this cytosine base in DNA, and after this transfer of CH3 onto this 5 carbon position, it will be termed as 5 methyl cytosine. Okay, so it will be producing 5 methyl cytosine in this case. Now if we look at the DNA methylation sites, as I've told you that these are very specific type of sites, these are very specific site of sites, they are called the CPG islands. Okay, so CPG islands, P means it's a phosphate, simply C cytosine, then a phosphate, so it's a backbone of phosphate, then another G, so that means again a phosphate, then a C, then a G. So that means we are having repeated sequence of CG repeats, right? So these CG repeats are hotspot sequences for DNA methylation, and the regions that are found in our genome consistent more using this or with this CG repeats are called the gene hotspots. Why they are called genetic hotspot? Because these are the regions for active methylation. Now as methylation can go on all the time in these regions, or these regions are much more specific for the methylation, there is a tendency of changing or modifying the gene expression from these sequences in our genome. So you can see here these are the spots that are responsible for silencing the gene, because usually the activity of DNA methylation is to silence the segment of genes. So it will inhibit the transcription of eukaryotic gene. That is the actual goal of DNA methylation. Now why we at all need this kind of gene silencing? Remember, we are having many genes, we are having almost all the same type or similar type of genes in all the different cells. Now cells of our brain is having the same gene that are present in cells of our liver, but we don't want all this gene to be expressed at a particular time. The expression of genes are tissue specific, We all know that, right? That's why we are having different or variation in function of a brain cell than the variation of function of a liver cell. So all of them are variation in the function due to this kind of tissue specific manner of gene expression, right? So silencing of tissue specific genes are important to prevent them from being expressed in the wrong tissue. That's a very very important concept, and for doing this we need to methylate some gene so that they cannot be expressed in a particular time in a particular tissue. So for that reason we are silencing all these genes. So gene silencing is important, and DNA methylation is playing the major role in this silencing. Now let us talk about the role of this methylase. As I've told you, the major role is to silence the gene so that tissue specific expression occurs all the time. And there are two different type of DNA methylase that we can designate. Usually the DNA methylase indicates, or it will methylate two different sequence. One is the C or cytosine, another one is A or adenine. Those methylase which usually methylate the adenosine residue is called DAM or DNA DAM methylase, and those one which usually methylates the cytosine residue are called DCM, right? So why these terms came? So this is cytosine methyl transferase, and this is adenosine methyl transferase, right? So that's why the name come from DCM and DAM. Okay, now usually they methylate at this 5 carbon position of the cytosine. So we have seen the picture of producing and converting into 5 methyl cytosine. Okay, okay, so if we look at here, this is depending upon which base they are methylating. But again, the methyl or methylation process occurs in a cyclic structure like that. So suppose this is a normal DNA without having any kind of methylation. So it's a very normal DNA which is just being produced, no methylation now. So we need a new type of methyl transferase to methylate this unmethylated DNA, because as it is not methylated at any time, so this is the first kind of methylation, and for the attachment of first type of methyl group to the DNA strand we need a new type of methylase. It is called de novo methylase. Why it is called de novo? Because it is starting from this methylation from the scratch, right? So DNA de novo methylase will come and methylate this C here, then the methylation is completed. Now as we can see that de novo methylase first methylates any one of the strands of this DNA. Now after that, when this DNA is resynthesized and the new DNA is synthesized from this old one, what will happen, one strand is having the methylation but the new strand is not having any kind of methylation, and that part is very very important, because that is to designate or differentiate between two different strands, the old strand and the new strand. This is a process of methylating only the old strand but not the new strand right after the DNA replication. But after some time the new strand will also be methylated. Now the second type of methylation that need to be carried out here is not by de novo methylase. This type of methylation can be carried out by less effective methylase. These are called simple maintenance methylase. Now these are the second type of methylase. This one is the first type, we have talked about it. Okay, so maintenance methylase will methylate the newly synthesized DNA strands so that we are having both the strands methylated. Now we are having two conditions. One condition is having one strand methylated, another condition is having both the strands methylated. Now if we are having only one strand methylated, we call that kind of DNA a hemimethylated DNA, and those type of methylation is called hemimethylation, because we are having only one strand methylated. On the other hand, if we are having both the strands methylated, we'll be calling it as a complete methylation. Okay, now this kind of complete methylation can be again demethylated, because methylation, if all the time it is getting methylated for this purpose, if it is methylated all the time, then that can create a problem. Then that gene can be silenced throughout the time, but we won't want that, right? So we what we want is the tissue specific manner and the time specific expression of genes. So suppose sometime we need this gene to be silenced, but now after some time we need this gene to be expressed. So in those conditions we must have removed this kind of methyl groups. So for removing this methyl group we need another type of enzyme. So I am changing my color, another type of enzyme that is called demethylase. This enzyme, so this is enzyme number 2 and this one is the enzyme number 1. So it is called demethylase. It will cleave this methyl group out of this position to get this unmethylated DNA into its original conformation, original form. Okay, so we are having two enzymes, methyl transferase or methylase and demethylase. Now methylase enzyme usually have two different types, de novo as well as maintenance. De novo methylates the unmethylated one, maintenance methylated the hemimethylated one. So this is the actual cycle of DNA methylation. Now as we can see here, this is the unmethylated form. Now this one is a hemimethylated form because we are having one strand methylated. But this one is fully methylated because we are having methylation in both the strands. Okay, but again I'm telling you, if these are the CpG island, because as you can see CNG repeat sequences, but the methylation always occur in the cytosine because there are DCMs or cytosine methyl transferase. And if there are A sequences, then DAM methyl is usually present in prokaryotes and DCM methyl is usually present in eukaryotes. So we are talking about eukaryotes, so that's why we have found this CpG islands here. Okay, now let's talk about the role of this methylation in the transcription purpose. Now I've told you that the activity of methylase is to block the transcription, is to halt the transcription so that the expression of certain genes are getting silenced, right? So it can achieve this goal by two different means. Now the first mean is to methylation of CpG islands may prevent or enhance the binding of regulatory transcription factors. Now what we know is this, there are some transcription factors or activator factors. For example, this one is an activator factor or transcription factor. Now in eukaryotic system, these transcription factors play a very important role for the initiation of the transcription process. Now for the initiation of transcription, these activator proteins or transcription factors must have bind to the enhancer region uh, present in the mRNA, right, which is to be transcribed, right, uh, present at the present at the DNA, which is to be transcribed, not mRNA. Okay, so but what in this case what is happening, that due to methylation, so you can see in usual normal situation, this is the DNA. Now onto this coding sequence, what we are having this activator protein to be attached to this promoter region, which is present right behind the enhancer sequence. Now as it present, it will send some signal which will recruit polymerase and transcription will go on. But due to kind of methylation onto this CpG island, what it does, it modifies this part of the DNA. As a result of this methylation onto this part of the DNA, this activator protein is no longer able to bind with this enhancer element. So as a result of unbinding of this activator protein to the enhancer element, it cannot provide any signal, so polymerase cannot call upon. So as a result, no transcription in this case. Okay, so this is a process of silencing the gene using methylation. Now another second process is also available, which is depicted as using this picture, is that the methylation inhibits the transcription is via protein known as methyl CpG binding protein or MBD, methyl CpG binding protein, right? Now this one bind methylated sequences. So now what we can see, that here it is a normal type of normal DNA sequence where we are having core promoter region from where the polymerase will sit and the reaction will start. But what happens here, due to the methylation, there are proteins recruited to the methylation stage. These are called MBD or methyl CpG binding protein. Now as these proteins are recruited, they are having higher affinity towards the methyl group, so they will attach with this DNA via this methyl group using covalent interaction. As a result, no further activator protein can come and sit here. So no further activator protein can come and sit, as well as no polymerase sequence can come and sit. So as a result of this process, they cannot start the transcription. And more importantly, this kind of MBD sequence attachment will recruit another type of protein in this situation. It is called histone deacetylase or HDAC into this situation, into this scene. So as they recruit HDAC into this position, now the role of HDAC is to silence the gene again, because the HDAC will allow this DNA to be wrapped around histone tightly so that they cannot be expressed. So now if they bring upon this HDAC, they will again deacetylate all these residues. As a result, they will start coiling onto the histone and won't be expressed anymore. So these are the two-way mechanisms that we can see conducted by the methyl transferase enzymes with the help of proteins like MBD and HDAC. Now this part of this this part is very very important. This is the crosstalk between the acetylation and methylation. Remember, now what happens in previous case? So we are having this green regions are the histone proteins, and this black thread-like structures are the DNA. Now suppose here are the histones are modified, so please don't make a complexity here, because the methylation or acetylation, all these things can happen in histone as well as in DNA. Now in this case we can see there is a acetylated histones here, so this one is acetylated, as this green color is acetylated, that means the histone protein is acetylated. Now also we are having DNA, some DNA regions which are methylated using methyl transferase. Now what it can do, it can recruit the ME or methyl CP2 proteins. Now after that it will recruit HDAC. Remember, in the previous picture we have seen that after the attachment of methyl CpG binding protein or MBD, it will recruit HDAC. So here similarly it will recruit HDAC. As it recruits HDAC, HDAC will act on, and because it is a histone deacetylase, so it will cleave this acetyl group and cleave it outside. As you can see, it is cleaving it outside. After that we are having no acetylation. Now remember, this acetylation always is telling us that to open the DNA complexes, and due to this deacetylation activity, this whole complex of histone DNA is tightly wrapped around into the compact structure. As a result, no further transcription event are possible. Okay, so this is a process which are initiated by this methylase activity, but it is enhanced by the presence of HDAC. So methylase can itself silence the gene, but if it itself cannot silence the gene, it can call upon other type of silencing agents like HDAC and recruit them so that they can achieve this task in great uh, easiness. Okay, so we are having this binding of methyl DNA, then recruitment of HDAC, and then deacetylation of the histone. So this is a crosstalk between acetylation and methylation. Okay. Now let us talk about a little bit about the gene insulators, which can also act as gene silencers. We have seen methylase to act as, or methyl transferase to act as a gene silencer, as well as HDAC to act as a gene silencing agent. But now we are going to see there are some sequences present in the DNA which can also act as gene silencer. They are called gene insulators. Okay, now these insulator are nothing but a segment of DNA that functions as a boundary between two genes. For example, here. So here it is gene A, here this one is gene B, and in between that we are having a boundary of DNA sequence. Some DNA sequences, but they are not any coding sequence, just a sequence. These are called insulators. Okay, similarly in this case also. Now an insulator is so named because it protects or insulates a gene from regulatory effects of the neighboring gene. For example, say here, for example, this is the gene A. Now if the gene A is transcribed, so it can produce this enhancer, right? Now this enhancer will be produced, so as a result of that it can go and attach to the insulator region. Now as it is bind to the insulator region, this both the expression of the gene A and gene B will happen in independent way. They will not occur in the attachment site. So suppose it's a very fine concept. Suppose this is a DNA molecule. In this molecule we are having, say, this is gene A, this is gene B. Now if these two genes are placed right after one another without being separated by any kind of DNA sequences, then here says a promoter sequence, here says it's the promoter region. So it will be recruiting the polymerase. So polymerase will come and sit, and they will transcribe whole gene. And what it does, it will transcribe both these genes together, as well as gene mRNA of gene A and mRNA of gene B, both of them together, right? So it will make both of the proteins together, protein component of A and B together, right? This is going to be the process. But if imagine if there is an insulator region present in the middle. Suppose this part is the insulator region. Now if this part present in this particular case, then what will happen? And if this region is blocked by a protein sequence, so let me take the protein. Here it is a protein sequence, a insulator binding protein. If it blocks the insulator regions by attachment, then what will happen? After that, even if the polymerase sit onto this promoter, it will only synthesize this A protein, but it cannot synthesize the mRNA of gene B, because there is a gap using insulator. So what insulator can do, it will block the dependence of both those genes, or linkage of this both those genes together. So as a result of the presence of this insulator, the gene A will be transcribed on its own and gene B will be transcribed on its own. So there will be independent transcription property, and that is achieved using these insulators. And we are going to see an important effect of this gene insulator to control or to regulate the expression of few genes which must be coming from our father, and also expression of few genes which must be coming from our mother, right? And this process is called genomic imprinting. So let's talk about it. So here it is, the role of insulator gene in the major scale is genomic imprinting. Why? Because this genomic imprinting is simply a phenomena in which the expression of certain genes is determined by whether the gene is inherited from the female or the male parent. That means there are some genes that are present in our body that can only be expressed from the paternal chromosome, and obviously there are some genes that can only be expressed from our maternal chromosome, that cannot be extracted from both the chromosomes, right? So these are some specific genes. For example, gene IGF2 is an imprinted gene, and those genes which are under control of genomic imprinting are called imprinted genes. So as an example, IGF2, which is an imprinted gene, can expressed only from the paternal chromosome. On the other hand, H19 can only be expressed from the maternal chromosome. For example, if you look at this picture, this picture will be clear to you that, for example, this maternal one, which is H19. So here we can see that two of the genes HGF2. So let me change the color. Here it is HGF2, which need to be expressed from paternal chromosome, but this chromosome in the maternal chromosome. But remember, all of our cells will have same number of chromosome and same type of genes. So both of them, maternal as well as paternal chromosome, are having both these genes, IGF2 as well as H19. So let's talk about this maternal one. So here it is, maternal chromosome is having gene IGF2 as well as H19. Okay, but what they are having, they are separated using an insulator sequence. And obviously there are some proteins that will go and sit onto this insulator to make a wall. So this protein is called CTCF, which is making a wall like structure. Okay, so we are having two genes, IGF2 here, H19 here. Okay, now here what we can see that activators, so there are some activator proteins which are present downstream here in this maternal chromosome. Now these activators are providing this protein. So this activator will come and it can sit onto this promoter, right? So imagine if this insulator is if the insulator is not present there, then what will happen? This activator can come and sit onto this promoter, or it may come to sit onto this promoter. So both of the possibilities can be there, and as a result of this activator attachment to the promoter, it will recruit polymerase, and the transcription of the gene will occur, right? So as a result, here what we can see, the activator will come and sit onto this promoter only. Why? Due to the presence of the insulator, this activator region cannot be attached to this promoter of IGF2. So as a result, it is ensuring that in this case only this H19 gene will be expressed, but not IGF2, because of the presence of this insulator and insulator binding protein. It will block the attachment of this enhancer sequence or activator protein with the promoter of IGF2, because what we know here, only we need the expression of H19 from the maternal chromosome, and this IGF2 must be expressed from the paternal one. So what we need here, we need to block the expression of this gene, and we can do this using this insulator sequence with the help of insulator binding proteins. Okay, similarly, if we look at the parental chromosome, so we are having again both these things, IGF2 as well as H19. Again we are having insulator at the middle. Now what we are having, again an enhancer further downstream of both the genes. So this can bind with both the sequence promoter of H19 as well as promoter of IGF2, but there is an insulator which is blocking this to happen. It is methylated. As this insulator is methylated and bound with some proteins, this enhancer region cannot be bring close together with this part. Why? Remember here, so here what we can see, this insulator is free. Just look at in this case, in the previous case the insulator is bound with protein CTCF, so that the bending, remember this enhancer need to be very close to the promoter sequence for recruiting the polymerase, right? Because we need to bring them. For bringing, we need to bend this DNA. Due to the presence of this protein, it cannot be bended so far that it can bind with the promoter of IGF-2. That's why the expression of IGF-2 is never there. But it can easily bend onto the promoter of H19. That's why the H19 expression is always on. But in this case, we don't have any protein which is bound with the insulator region, so bending can be easier. But on the other hand, what we are having, methylation. Now the methylation can be obtained in this H19 gene. Even if this enhancer can be bound here, it can easily be bound here, it can easily be bound here, both this case. But due to the methylation that is present in H19 gene, even if the enhancer bound, even if this activator protein bound with this promoter sequence, it won't recruit polymerase. So the expression of H19 gene will be halted, and only the expression of IGF-2 will be on, because there is no methylation present in this IGF-2. So it ensures that this paternal chromosome will express this IGF-2 but not H19. Similarly, in this maternal chromosome can only express H19 but not IGF-2. So these are the process of blocking, or of having what we can say gender based or sex based expression of some genes, which is very very important, because we need some traits coming from our father only, we need some traits coming from our mother only. Okay, so that is a very very important part. Okay, so till now we have talked about the DNA methylation studies. Now let us talk a little bit about the protein methylation, because I have told you that this methyl transferase can methylate both these things, DNA as well as proteins. Now protein methylation, again it's a covalent modifications, and usually the protein methylation is also specific, just like the DNA methylation, because in DNA methylation we have seen the methylation always tends to occur in the cytosine as well as in adenosine. In this case of protein methylation, usually they methylate the carboxyl group of glutamate, leucine, or isophenyl cytosine, so whatever, majorly glutamine and leucine, and the carboxyl group of them. And they can also methylate lysine residues. And in histone, whenever we talk about protein methylation, we are more interested about the histone methylation, because histone is the protein uh, which is a which is helping to DNA to wrap around itself, right? So we'll talk about this histone methylation. And histones are having many lysine residues outside, because it is required that lysine or positively charged lysine residues to be wrapped around, to be wrapping for the wrapping of DNA, which is negatively charged. So it is having more lysine. So as this methyl group can be attached to the lysine residues, it is much more easy to methylate the histone proteins. Okay, so let's look at the picture. So here it comes. It can methylate both lysine as well as arginine residues. Both of them are of this positive charge. So here it is the arginine having this NH2 groups. In this case, this is the R chain of the arginine. Now methylation can be presented onto one of this chain using PMT. We can say it's a methyl transferase using another supporting enzyme adomate. Okay, now this one methylation or monomethyl transfer is possible. After the single methylation of arginine, it will be called as monomethyl arginine. Then it can have second round of methylation. The methylation of second round can be of two type. One is that methylation of the same nitrogen, so you are having two methyl group attached to the same single nitrogen. Here we'll be calling as the asymmetric dimethylation. But in this case we can see it is having a symmetry, because if we draw this, it is having a symmetrical sequence, so we'll call it a symmetrical dimethyl arginine. So it can produce monomethyl arginine as well as dimethyl arginine. Now the production of dimethyl arginine can be of two different type, asymmetrical and symmetrical. Okay, so here it is, the asymmetrical one onto the same binding region. This is the symmetrical one, which is onto the two different nitrogens. Okay, and this one is the methylation of lysine residue, so it is the lysine. Now HMT or histone methyl transferase will come, and obviously with the help of adomate, it will attach this methyl group to one of the nitrogen. Again here it is a CH attached to here. It is forming this or converting this lysine into monomethyl lysine. Then again the second round of methylation is also possible. It is called the dimethyl lysine. Now the methylation for lysine, the second methylation of lysine, is only one type, which is symmetrical always. It is dimethyl lysine. And a third layer of methylation is also possible, is it will be called a trimethyl lysine now. Okay, so this type of methylation can be possible, and as you can see, lysine can hold on to three different methylation at the same time. So that is creating lysine a very hot spot for methylation. Now, as we've seen that the methylation of DNA as well as the methylation of proteins, now let us combine all of these things or couple this histone methylation with the DNA methylation, and what are the effects that are brought about by both of this methylation happening together. Now, say here it is the methylation events. It can be transferred from one region to another region. That means the methylation in DNA region. So if I draw this structure of, say, this is the DNA, this is the methyl, so this is again histone, so like that. So these are the histones. So let me, this is actually not a good drawing of histone. So it is two histone and three histones here and the DNA sequences. So if they are methylated, for example, so here it says methylation can be in the DNA. Now if there is a methylation in the DNA, it can be transferred, or not transferred, but this methylation from DNA can instigate to methylate histone, or sometimes methylation in histone can help us to methylate nearby DNA region. But again, provided the fact that these histones and DNA are present in close vicinity, right? This is very, very important. Okay, so the transfer, not the transfer, but also the effect of methylation can bring about from DNA to protein and sometimes from protein to DNA, right? Here protein means histone in all these cases. Okay. Okay, so let's look at the first kind where the methylation event will be done from DNA to protein. So here it is, the DNA is already methylated. In these red colored regions are the methylation, so they are already methylated, and now MBD or methyl CpG binding domain or CpG binding protein, MBD will come and attach to this part, and we know that MBD can recruit another type of deacetylase enzyme or histone deacetylase or HDAC enzyme. Now this is not a single enzyme, there are some enzyme complexes, and we are going to see in the future size what are the enzyme complexes. So they will recruit this HDAC complex here. Now this HDAC complex will methylate histone tails. Now we are looking at methylation from DNA to protein. So now previously the DNA was methylated, but after recruiting HDAC by MBD, it will recruit the methyl group to the histone tail. So now the histone tails will be, are going to methylate. Okay, histone tails will be methylated in all this case. Okay, now in the second kind, so this kind of methylation is indirect because we need the presence of MBD and HDAC for the completion of the process. Now the another type of the same type of DNA to protein methylation can be of direct type. And the direct type, we need only one type of complex, which is MBD complex with a protein called ACT. Now this type of methyl transferase enzymes directly recruit a methyl group to the histone tails. They can directly recruit this group, direct recruitment of this methyl group to the histone proteins. So due to this direct recruitment onto this histone proteins, it can also methylate them, right? So these are the two types, but again the methylation from DNA to histone. But the total different second type of uh, methylation event is can be from the histone to DNA or protein to DNA. So here it says the protein or histone tail, which is already being methylated here. Now it will recruit a protein called SF1, SP1. Now this SP1 can further recruit another protein called DNMT or DNA methyl transferase. Now this DNA methyl transferase are having two different domains. One is the attachment of SP1, another one is the methylation domain. Now using the methylation domain, it can methylate the nearby DNA sequence by recognizing the methyl group that is present on the histone tail. So why it is important to methylate DNA if a histone is methylated, or why it is important to methylate histone when the DNA is methylated? Because methylation in histone and DNA recruits more or less the similar effects. Okay, so and the effect is the gene silencing. So if we need to bring about the gene silencing, the methylation of only DNA is not enough. We need to methylate the histones also, because for the compaction of the DNA, for the unavailability of the DNA, it must be wrapped around the histones, right? Okay, so that's the process. Now this kind of attachment of methyl groups can lead to the silencing of chromosome. Now what we know for eukaryotic system, there are two type of chromatin. One is the heterochromatin, another one is the euchromatin. Now what we know, this euchromatins are the type of chromatins which are expressing or expressive all the time. and heterochromatins are those one which cannot be expressed all the time. So here it says in the left hand side we are having the heterochromatin. So heterochromatins are usually those we don't need the expression of this one, but we need the expression of euchromatin. But in certain conditions we also need the expression of this heterochromatin. We need to block the expression of this heterochromatin, because there are two types of heterochromatin: obligate heterochromatin and facultative heterochromatin, right? So what are, which are facultative heterochromatin, they are not, we are not going to talk about, but some of the obligate heterochromatin, so some of the facultative heterochromatin can actually uh, uh, can be expressed sometimes. So we need to stop the expression of those facultative heterochromatins sometimes. So for that reason we can silence the heterochromatin region. And euchromatins are the chromatins, or rest of the chromatins which are being transcribed and translated all the times, so we may need to silence them many occasions. Okay, so let's look at the heterochromatin silencing. And in this case we are having the DNA, and along with we are having the histone attached here. So H3 is the histone 3 which is attached here. So what we are having, the DNA which is having two sequence of lysine 9 and lysine 14, which are both acetylated. Now what we do in this case, we need to deacetylate them, and we can deacetylate because acetylation brings about the open complex of DNA histone. So now we are having DNA open. So if we deacetylate them, it will lead to the conversion of open complex into the closed complex. So what we can do, we can add the lysine deacetylase, which is the HDAC or histone deacetylase, which will deacetylase this K9 and K14 of lysine. And then what we recruit sub 39H1, which is a methyl agent or methylation agent, which is also called methyl transferase. Now this sub 39H1 will be added, and they will methylate this particular position of K9. So now previously which was K9 acetyl condition, now it is getting methylated after the cleavage of this acetyl group. Then we'll recruit HP1 molecule, which will be bound to this particular site, and keep on adding HP1 molecules so that it will wrap this DNA around the histone molecules so tightly, so that all of this euchromatin region, heterochromatin region are blocked for the expression, right? So what we can see here, sequential events and crosstalk between acetylation, methylation, as well as the HP1 self association for this purpose. Okay, so these are the major three key players for the silencing of heterochromatins. Okay, but for the euchromatin, what we can see, for the euchromatin we are having again the DNA sequence are released. Now we need to coil them. So for the coiling we must deacetylate whenever the acetyl group is present. So in this case we recruit the E2F factor first, then it will be recruiting S sub 39H1 and HP1. So it is called the RB complex or retinoblastoma complex, right? It's a tumor case, so we call it retinoblastoma complex. Now this complex with RB sub 93H1 and HP1 will be recruited, and they will go and they will be attached with this position of the histone tail, and it will methylate the histone tail. So here it comes the methylation at K9 position of the histone tail, and then what will happen, self recruitment of HP1 and self association of HP1 protein, just we have seen in case of heterochromatin. So they will recruit HP1, more and more HP1, and then they will be attaching or wrapping the DNA onto the histone, so that regions of DNA are being uh, blocked for the expression or are being stopped for the expression. Okay, so these are the two processes of gene expression studies that we have seen. Okay, so both of them are the gene silencing events for heterochromatin as well as for the euchromatin. Okay, so that's in a sense of methylation, and I hope that's helpful. Thank you.